Good to have you on Planetterrian Daily, episode 49, coming to you on April 23rd, 2026. Let's see what the latest research is telling us. Supplementing a specific polyunsaturated fatty acid restored visual function and reversed cellular aging signs in the retinas of older mice. Scientists at the University of California, Irvine targeted the ElevenLabs gene, often called an aging gene, in experiments with older mice. By restoring levels of certain long-chain polyunsaturated fatty acids in the retina, they saw clear improvements in the electrical responses of retinal cells. The treatment also reversed molecular markers of cellular senescence, the state where cells stop dividing and contribute to tissue decline. This suggests that a precise lipid supplement aimed at these specific fatty acids could prove more effective than using broad-spectrum DHA supplements alone for supporting visual acuity in aging eyes. The work highlights how lipid metabolism changes with age and offers a focused biological target rather than a general nutritional approach. It is still early-stage research in mice, but it adds a mechanistic layer to how we might one day intervene in age-related vision loss. While we are on the theme of precise molecular interventions, let us shift from restoring sight in mammals to protecting an essential food crop from a rapidly spreading pathogen. Researchers in Sweden have synthesized a peptide that selectively attacks Phytophthora infestans, the microbe responsible for potato late blight. The molecule leaves other plants unharmed, which is a significant advantage over broader fungicides. Late blight has long threatened potato and tomato crops, and climate change is expanding the disease geographic range. This new peptide offers a potential tool that could reduce ecological disruption while safeguarding yields in vulnerable regions. The approach demonstrates how synthetic biology can deliver highly targeted solutions to long-standing agricultural problems. It is a promising development for food security without relying on methods that affect non-target organisms. Staying with clever molecular design, scientists are also rethinking how we find new drugs from the very beginning. A recent review details how fragment screening is changing early drug discovery by identifying small chemical pieces that bind weakly but specifically to disease targets. Chemists then grow these validated fragments into larger, more potent molecules rather than relying on traditional trial-and-error methods. The technique focuses effort only on fragments with confirmed binding, which trims years off the early-stage pipeline. This structured approach replaces much of the guesswork that has slowed pharmaceutical development for decades. It represents a genuine advance in chemical biology by giving researchers a more rational starting point. From designing molecules in the lab to designing better ways to observe the living world, a new European framework aims to bring order to long-term ecosystem data. The ELTER framework of standard observations creates a harmonized system for collecting comparable data across Europe's varied ecosystems. It spans multiple research fields and allows scientists to track environmental trends over decades with greater consistency. Previously inconsistent methodologies have made it difficult to compare results between sites and over time. This new standard observation set addresses that problem directly and should improve the reliability of long-term ecological monitoring. Coordinated data like this will strengthen our understanding of how ecosystems respond to climate and land use pressures. Speaking of how environment and population shape biology, an intriguing new study looks at what actually drives mating systems in birds. An international team analyzed 261 bird species and found that adult sex ratios and other demographic factors primarily drive variation in mating systems, parental cooperation, and sexual ornamentation. Statistical models suggest that population structure sets the evolutionary stage rather than the behaviors shaping the demographics. This challenges the common assumption that mating behaviors come first and then influence population dynamics. The findings reframe how we think about the evolution of reproductive strategies across species. It is a reminder that demographic realities can exert stronger selective pressure than we often credit. From those insights into wild behavior to deliberate genetic editing in the barnyard, researchers have taken another step toward chickens that lay therapeutic eggs. A team at the University of Missouri has refined gene editing techniques in chickens to produce therapeutic proteins directly in their eggs. The work builds on the existing use of eggs for harvesting antibodies and expands the possibilities for scalable biologic manufacturing. This platform could offer a cost-effective way to generate future biologics at larger volumes than some current methods allow. It demonstrates how applied genetics can turn agricultural animals into efficient protein factories. The advance is another example of biotechnology repurposing biological systems for human therapeutic needs. While we are thinking about how cells allocate their resources, there is a fascinating basic biology finding that reveals bacteria keep surprisingly large protein stockpiles on purpose. Most people imagine cells as tidy factories that make exactly the proteins they need and quickly discard any extras. In reality, your cells and the bacteria in your gut maintain surprisingly large stockpiles of certain proteins even when it appears wasteful. Right now, as you sit listening, every bacterium in your gut and every one of your own cells is constantly deciding how much of each protein to keep on hand versus degrade. These reserves act like emergency buffers that allow rapid response to sudden changes in nutrients, temperature, or stress. A single E. coli cell can hold up to 10,000 copies of one regulatory protein, even though it only needs a few dozen at any moment. That overabundance looks inefficient until you realize the extra molecules buffer against random fluctuations and allow the cell to flip its behavior in seconds instead of minutes. The strategy trades apparent waste for speed and robustness. Understanding these hidden stockpiles is already changing how bioengineers design microbes for drug production and could one day help us fine-tune human cells that lose responsiveness during aging or disease. The next time you hear that a cell wastes energy, remember it may be investing in speed and robustness instead. Watch for future research that maps exactly which proteins different tissues stockpile, because those inventories will likely become new targets for therapies that restore youthful cellular responsiveness. A separate life cycle assessment has shown that including soil organic carbon dynamics raises the estimated emissions from milk production substantially. When researchers accounted for freeze-thaw cycles in northern climates and other soil carbon changes, the footprint increased by 41% over standard IPCC methods. Current accounting approaches can therefore underestimate the climate impact of dairy systems in meaningful ways. The result underscores the importance of refining how we measure agricultural emissions, especially in regions with distinct seasonal soil processes. It is a useful reminder that life cycle assessments continue to evolve as we incorporate more complete environmental data. From the hidden impacts of what we eat to the hidden impacts of what we watch, our final story today looks at patterns of early adult content exposure. Researchers identified 3 distinct trajectories of first exposure to sexually explicit material in a large observational study. Beginning a regular habit at a younger age was correlated with higher rates of mental health difficulties in adulthood. The work adds nuance to ongoing discussions about how the timing of exposure may relate to long-term well-being. Because it is observational, the findings show association rather than direct causation, but they highlight patterns worth further investigation. Today's discoveries remind us that incremental advances in basic biology, from lipid restoration in the eye to smarter ways of monitoring ecosystems, quietly expand what is possible for both human health and planetary stewardship. Before we go, keep an eye on how these mechanistic insights into cellular resource allocation might influence the next wave of microbial engineering studies. That's Planetterrian Daily for today. If you enjoyed this, a rating or review on Apple Podcasts or Spotify really helps new listeners find the show. I'm Patrick in Vancouver. Thanks for listening, and I'll see you tomorrow. This podcast is curated by Patrick, but generated using AI voice synthesis of my voice using ElevenLabs. The primary reason to do this is I unfortunately don't have the time to be consistent with generating all the content, and wanted to focus on creating consistent and regular episodes for all the themes that I enjoy, and I hope others do as well.